Welcome to Infectious Diseases. You are stepping out of the microscopic world of pure cell biology and into the macroscopic world of human survival. This unit is about a biological arms race. To understand how a microscopic invader survives and multiplies, you have to know exactly how it exploits our cellular machinery. We will connect your foundational knowledge of transport systems, cell structures, and genetics to real-world epidemiology. You will see how environmental, social, and economic factors are just as critical to disease distribution as the biology of the pathogens themselves. Before we can fight these diseases, we must define the enemy and understand how they spread. A pathogen is simply a biological agent that causes disease. In this unit, you will encounter bacteria, viruses, fungi, and prototictists, each requiring entirely different strategies for control. But how do these microscopic agents jump from host to host? We call this transmission. It might be airborne droplets from a cough, contaminated drinking water, or a biting insect vector. Break the transmission chain, and you stop the disease. In the exam, you will frequently be asked to deduce the most likely transmission route from a short description, so linking the pathogen's biology to its spread is essential. The Cambridge syllabus requires you to master 4 specific, globally significant diseases. Let us look at the first 2. Cholera is caused by a waterborne bacterium called Vibrio cholerae. It secretes a toxin that permanently activates cellular pathways in the gut, triggering severe dehydration. Malaria, on the other hand, is caused by a complex prototictist called Plasmodium. It relies on the female Anopheles mosquito as an insect vector to infect human blood. You will need to know their exact binomial names, their life cycles, and how social factors drive their global distribution.
Next, we examine 2 diseases that devastate the human immune and respiratory systems. Tuberculosis is an airborne bacterium that destroys lung tissue. In the exam, you will link the biology of these pathogens to their symptoms. For example, TB bacteria have a waxy cell wall that allows them to survive right inside our immune cells. HIV is a retrovirus that spreads through bodily fluids. It systematically dismantles the very immune system meant to fight it, specifically targeting your T helper cells. Understanding this cellular detail is exactly how you secure the top marks. Once infected, our primary pharmacological weapons against bacterial infections are antibiotics. You will learn exactly how drugs like penicillin disrupt bacterial cell wall synthesis, causing the bacteria to burst. You also need to explain why they are completely useless against viruses, which lack these cellular structures. But there is a modern crisis. Through random mutation and natural selection, bacteria are evolving resistance to our drugs. When we overuse antibiotics, we inadvertently create superbugs. You will map this evolutionary process step by step for the mark scheme, starting from a random mutation all the way to a resistant population. So, as you work through this unit, you will learn to identify the exact pathogens behind major diseases, explain their transmission routes, evaluate global control methods, and outline the critical mechanisms of antibiotic action and resistance. This is where your biology knowledge saves lives.